Good afternoon, everybody. Today, I will be talking about the effect of applying different nitrogen sources to the root dynamics of tomato plant. And I will be talking about a bit of root morphology, change on rhizosphere pH, and as well as the associated bacteria. But before explaining the experiment and the main results, I would like to introduce what kind of nitrogen source I will be talking about. In my group in the Forschungszentrum in Jülich, the alternative biomass group, we have been working a lot with recycling or testing the nutrient use efficiency of recycled products. So today I will be talking about recycled nitrogen sources. And we believe the better recycle of the nutrients and improving the use efficiency of these nutrients can help us to have resource autonomy in Europe. So one of the examples we use, it's struvite. It's a precipitated that can be recovered from different waste streams that can be, for example, manure or wastewater. And the aim is to close the nutrient loop by recovering it and then recycling it as a fertilizer. It's a precipitated of ammonium, magnesium, and phosphate and has about 6.6% of ammonium. And there have been going on a lot of studies on the effect of struvite on plant performance and root dynamics, but mainly on phosphorus. And during my PhD, I also focused a bit on that. And we analyzed, I tried to find those root traits and also which plant species will increase the use efficiency of the struvite as a phosphorus soil in that case. And for example, we realized, or we investigated that narrow leaf looping could increase the use efficiency of struvite to have the same result that using the high soluble potassium phosphate due to the, as Hans Lambers explained in his talk, the high release of exudates of carboxylate that this plant has and help to mobilize it. But coming back to the nitrogen here in this session, I also wanted to check what would be the effect of using struvite as a nitrogen source, and therefore I compare this with an organic fertilizer that it's in the form of readily available amino acids, and it is commercially used as a, let's say, recycled nitrogen source somehow. So when I study the nutrient use efficiency of these recycled products, like the same of any fertilizer, I try to investigate all these, or take into account all these plant-rhizosphere-soil interactions. And if we have been hearing during the talks all those days, this can be quite complicated. And the factor of applying the fertilizer as inorganic source of nitrogen or organic will have a tremendous effect on different traits, and the nitrogen availability will be different, and it will affect all the factors like the soil pH and vice versa. The soil pH can also affect the availability of the nitrogen in these nutrients, will also affect the root architecture and the microbial community and so on. So I try to summarize all these interactions in the three main sections that I will be talking about in my talk, where I focus on what will be the effect of these two nitrogen sources applied on the plant performance and nutrient uptake or nitrogen uptake, and also what would be the effect on the root morphology, mainly talk briefly about the root architecture change, root physiology, mainly about change in the rhizosphere pH dynamics, and then I will move to what is the effect of this and the nutrient turnover for these products, and what is the effect of the microbial community, what roles they play in all of this. Briefly, the setup of the experiment, we grow the plants in these rhizotrons that allow us to visualize the root growth and also to measure the dynamic change of the pH. And we have the organic fertilizer, the struvite, as inorganic recycled nitrogen source, and also the known nitrogen. And we have three different time points: the beginning of the experiment when we sow the seeds, and then we have to harvest the time point 2, that it's the first harvest 20 days after sowing, and the third time point 34 days after sowing. We also have always the tomato plants and rhizotrons with no plants grown on them. And the questions, for each time point, we measure different things. Of course, we measure the chemical composition of the substrate and also take the samples for the microbial community analysis. And in time point 2 and 3, where the plants were already growing, we also analyzed the plant performance above ground and below ground with root architecture. The only small change in the last time point, we measure the activity test of the microbial community as well. So with these, I aim to answer 4 main questions that were: what is the nitrogen source that we apply effect on shoot, and also on the root architecture, what is the answer effect on the pH dynamics, and how all of these changes in the root morphology and the pH dynamics can affect the nutrient turnover of these 2 sources, and then finally, what is the effect on the microbial community on all of that and how it's affected. So starting with the result for the first 2 sections, where I will talk first about plant performance and mainly about root architecture and root physiology or change in the pH dynamics. So when we apply the struvite and the organic fertilizer, I might expect it to have also very good results with the struvite, because very summarized, I expect that this is what will happen. However, with the organic, we will need to have very simplified, let's say, an extra step. The nitrogen in the organic form will need to first go to ammonium and then to nitrate. But as you can already see in the picture, the organic fertilizer produces higher yields within the tomato plant, that you can see also in the graph for the leaf area. In the second time point, it's even more clear that the organic fertilizer performs better than the struvite. And if we look into the roots, we also observe these are different time points, because as I mentioned, with the rhizotrons, we could follow up the root growth during the whole experiment. So at different time points, we also observed that from the very beginning, the organic fertilizer produced higher visible amount of root length than the struvite and also the no nitrogen application. This is also true for the specific root length and also the root surface area. I cannot show here all the results, but it is showing the first main result summarized that the organic was doing better than the struvite for somehow in the plant performance. If we move to the plant physiology or root physiology bit, let's check the pH dynamic change. For that, you can see here the rhizotron, and this white stripe you see here in the middle, it's so-called optods, and this is a pH sensor that allows us to measure dynamically the pH change in the rhizosphere. So for that, we use hyperspectral cameras that allow us to transfer the information to a software, and by a calibration, we could have the value of this picture into a real pH value. So you see here the same rhizotron treated with the tomato plant treated with the organic fertilizer at different time points. And in the orange circle, that is what I call near rhizosphere, or well, near the root, you see change in the color going a bit more to the purple and then coming back to the orange. And this was not observed in the area a bit far from the root. And this is showing change in pH, in this case, alkalinization. So if you see here this simple graph where we have an increase of pH and then a decrease that it's correlated with this color change. So that was indicating that we were able to measure a small alkalinization for a short period of time. And coming back to the nitrogen, we thought that might be related with an uptake of nitrate that will alkalinize the rhizosphere in that exact moment. So to really check if this that we hypothesized might be true or not, we needed to measure the concentration of ammonium and nitrate that will be in the substrate. And as well, after that, related to what the microbes will be doing. So for the nitrogen dynamics, we observed that the nitrogen source that we apply affected the nitrogen turnover. This is a very full table, but I want you to. I will show only the data from the tomato plants, where we don't have plants growing. So this summarized table. If we look only with the organic fertilizer, these are the measurements of the ammonium or nitrate concentration in the substrate at the beginning of the experiment, the first and the second harvest. And you can see that with the ammonium concentration in the first harvest already decreased from the beginning of the experiment because part of this ammonium has been already mineralized to nitrate, where we have an increase of the concentration. You can see here from non-measured to here. And in the third harvest, even more of this ammonium was not measured in the substrate, but also we didn't observe and keep increasing the nitrates. That might be an indication that this nitrate was actually being taken up by the plant. That might be related with the measurements of the optots that I just showed. And if we look at the struvite effect, what we observed was something totally different, meaning that with the struvite, the concentration of ammonium kept increasing during the whole experiment, but we didn't measure any nitrate actually in the substrate. So what makes me think that what was happening is when we applied the struvite treatment, this step was not taking place. And with the organic, high amount of the organic nitrogen was fast transformed to nitrate. But surprisingly, we will expect then that the plants will be recovering much more nitrogen from the organic than from the struvite, maybe. But the difference in the nitrogen recovery were not that big between the two fertilizers. Still, we have with the organic around 25% of the amount of nitrogen that we apply recovered by the plant. But from the struvite, we still have around 20%. But interestingly, what we measure in the substrates at the end of the experiment, from the struvite, it's almost all the rest that it's not taken by the plant. So the plant was able to take this ammonium, and the rest was kept there in the substrate. However, with the organic, the plant that took around 25% of what we apply, but the rest was not measured there in the form of N-min, so somehow it's still in the form of organic or used by someone else. And that means that the recovery from the organic was still a bit better than the struvite, but this mineralization and transformation from ammonium to nitrate was not only to be used by the plant, let's say, so someone else might be taking a role. And I want to mention that this effect of the plant, I didn't show here all the data in this table, but when we don't have plants growing, with the organic fertilizer, we still see this mineralization of ammonium to nitrate with the organic fertilizer, but it occurs much slower than when we don't have the plant. So the next step was who might be then playing a role there and eating this nitrogen. And I'm not an expertise on all this work of microbial community, but I get in contact with the University of Ghent, and Dr. Oliver Grunet helped me to do, I helped him to run all these microbial analysis. We took into account the area where we collect the soil to measure the microbial community, let's say near from the rhizosphere and far, as I show you in the pictures of the pH dynamics, and then we run from the soil taken in this area, the different analysis. Very, very summarized. Those are the results for, let's call it, far rhizosphere. I don't know, you might have a better term for that. But this is the principal component analysis for the relative abundance. And what we observe is the fertilizer, the presence or absence of the plant on the harvest time had an effect on the relative abundance that we found in the far rhizosphere. But when we run the permanova analysis, after that to check what was really the presence of the microbes there, there are more information here, but if you focus only on the colors, you can already see that there is a separation between what the organic and struvite with the no fertilizer, so difference between these two, and also the solid symbols at this time point 3 and the hollow symbols at time point 2, and this is also showing a clear separation of the microbial community between the time points. So fertilizer has an effect, but mainly the time point where we harvest the microbial community. And when we move to the near or close rhizosphere, again, the relative abundance has a significant effect of the fertilizer applied, but mainly I will say that it was significant that become stable over time because we have here the significant much smaller in time point 3. It might indicate that the microbial community get more specific over time. And actually, when we check the Bermanova analysis here, this effect of the differentiation between fertilizers, it's not seen anymore. So that's not that important. We still see the effect of the separation between the time points. And very briefly, to finish my last slide, we also measured the activity of the potential ammonia oxidation rate. And interestingly, it gets decreased in the area near the root, and it's in comparison with far from the roots. That might be an indication, maybe the plant with some nitrification inhibition or some competition, but we could not go farther on this analysis. So to summarize, I know it's a big bunch of different data, but to try to summarize, the nitrogen source that we apply, of course, affected plant growth and the morphology of the root. The straw then show lower nitrogen availability than the organic fertilizer, and this short-term pH in the rhizosphere might be related with the uptake of nitrate when we have the organic. And I will say that the root location near our rhizosphere and the plant developmental stage, or the time point where we did the harvest, has the highest variation in the microbial community. So there will be next steps to clarify all this information, because maybe the straw fertilizer, in that case, it's more affected by abiotic or physical factors, or we maybe lack the right microbial community and we need to find a more favorable microbiome for that fertilizer, or investigate really which microbial community explain this change in plant performance, meaning we don't know if it's the microbial community affecting the pH that will affect the plant performance, or vice versa, the other way. So with that, I would like to thank all my colleagues from Jülich, and of course the people from Ghent University to introduce me in the amazing world of the microbes, and Vicky Temperton that helped me to try to interpret all this data. Thank you for your attention.